Hi, my name is Cecily Cidirol. I'm one of the grad students that's going to be presenting today, and today what I'm going to be doing is um, methods and techniques of my own research. So, kind of going over some things we went over in week 3, and also sharing some newer ones that we haven't gone over. So, just some background about what my research is, so we can have a better understanding of the tools that we're using. Um, I do research on Cyclospora caintensis, and it's a protozoan parasite that causes cyclosporiasis. And it's a diarrheal disease that's worldwide. It can only cause infection within a human host, so there is no animal model that we can use to further research. Um, it's also highly unlikely to spread from one person to another, like directly, because the parasite needs to sporulate in the environment, and then that's the infectious stage. And so it's shed from someone unsporulated, and then it sporulates in the environment. So that once someone ingests a sporulated oocyst, which is the form of Cyclospora caintensis, that's what causes infection. It's also shed by a infected person's stool, and it's known to be spread by the consumption of fresh produce or water that's been contaminated with these sporulated oocysts from the parasite. So, what my research focuses on is the main purpose is to determine the prevalence and transmission of Cyclospora in fresh produce growing regions of Lake Okeechobee, South Central Florida. Some other objectives of this research is to determine if flow cytometry, which is something we're going to go over in a little bit, is effective in sorting the Cyclospora oocysts from environmental samples. So the oocysts are again the form of the parasite, and in the environment they sporulate, and that's what people ingest to become infected. And then we also want to determine if ImageStream, which is another tool we'll look at into further in the presentation, is effective in visualizing cyclosporosis from environmental samples. And a fourth major one is developing a target sequencing method of nuclear and mitochondrial markers on environmental samples using the Oxford Nanopore Mini Ion, which is another tool we'll look at. So first one that kind of covers our first objective, my first objective of research, which is trying to look at the prevalence of Cyclospora in the environment, is we use qPCR. So qPCR is a quantitative polymerase chain reaction. That's what it's short for. And it's a technique used to measure the amount of DNA or RNA in a sample. So this technique uses a special type of enzyme, which is polymerase, to amplify the amount of DNA or RNA in the sample. And the polymerase enzyme creates copies of the DNA or RNA, and also in my case, it's DNA, by adding new nucleotides to a template strand. And the qPCR uses these fluorescent dyes. So it's these probes that are specific to in my case, to the cyclospore 18S region of the DNA. So these probes are trying to amplify that part, and it's amplifying that part of the sequence to identify whether the cyclosporosis are there or not. So, well, the DNA, if the DNA is there. And it also has to use primers, use primers and probes to amplify the DNA, and then it uses the probes to fluoresce and identify whether that sequence is there or not. It also tells you in kind of, well, it tells you the amount of the concentration of DNA that's there also, because the way you read this is it's by these numbers called CT values, and what it's doing is it's reading where in the cycles of the qPCR that it's amplifying. So if it amplifies later in the cycles, then it has a way less concentration rather than amplifying sooner. So there is multiple cycles in here, and it goes through multiple different phases in order to amplify. And it's also trying to see whether that sequence is there or not. I hope that makes sense. But qPCR is a highly sensitive and specific technique that can be used for a variety of different applications, including gene expression analysis, pathogen detection, and genetic testing. And it's widely used in biomedical research, clinical diagnostics, and environmental monitoring. So another instrument we use that was my second objective in my research was to figure out if flow cytometry was effective or not. So what we use this machine for is to sort cells. So this machine can sort cells using size, shape, and fluorescent properties. And it can be used to analyze a large number of cells quickly, depending on the sample. And it's used in a variety of different applications. It's least used in environmental samples, as I've learned. So it's not too quick for my kind of samples, just due to the type of samples that I have. They're very large. The particles are very large compared to what it would usually be used for. It's especially used in cancer research, and it's actually in the cancer center at the U of A. But this technique can be used to study cell differentiation, apoptosis, cell cycle, and cell signaling pathways as well. But it's a very helpful tool for understanding the biology of cells, and it's widely used in research, clinical diagnostics, and drug discovery. So this machine is the ImageStream, and what we use this for is for imaging. This kind of goes hand in hand with the flow cytometer, because you can actually, well, before we get into that, this is what we used to actually visualize the cyclosporolosis. We were trying microscopy at first, but it was just not very efficient, and it actually took a very long time, and it just felt like we weren't really capturing anything. So we actually noticed that this ImageStream machine was at the cancer center as well. And it's very alike as the flow cytometer. So you can actually put your sample from flow, all the cells that you got, you could put it in this machine, and then you can capture pictures of the sample. But as you're capturing pictures of sample, you can actually also describe using the same things, fluorescence, size, and shape, of what pictures you want from that sample. So it is a lot like the flow cytometer, except you're actually taking images of it. And this right here is images of a positive control from our cyclosporolosis that we got from the CDC. Another tool we use is PCR. So we did look at qPCR, this is now PCR. And the difference here is this is just amplifying the DNA. So we can have a sample that has a higher concentration of DNA that we can further use. And in our case, we use it in genome sequencing. So genome sequencing is, well, let me go back to PCR. So PCR, there is kind of a little bit of a struggle with the cyclosporolosis samples because they are environmental samples. So there's not a high concentration of it in the samples. So we're actually trying to further a method to gain highest amount of DNA concentration as we can using PCR. So we've been troubleshooting a couple different ways of even doing PCR off of PCR products that we had. So basically, we would put the DNA into this machine using different primers, and nucleotides and different polymerase enzymes. to amplify the DNA. And then from there, we try to do another PCR from that to see if we can optimize the amount of DNA that we can get. But yeah, it's just very difficult because environmental samples, there's very low concentrations. And that also goes hand in hand with genome sequencing. So we use PCR so we can have the most amount of DNA that we can get and use it to do genome sequencing. Genome sequencing we use for a couple different things. Right now, the research that we're doing is trying to find a good methodology so we can use it as a second confirmation that it is Cyclospora that we're seeing. So basically with genome sequencing is we can actually prepare DNA and load it onto this device here. This is actually a more newer technology that people are using. It's called Oxford Nanopore Technologies. And you put the DNA in this device and it can be used to do whole genome sequencing. And there's actually several advantages to this rather than what's normally been used. Is it includes long read lengths, real-time data analysis, and portability. So what this means is you can actually see the data that you're getting as it's running. And it can run for 24 to 72 hours, but you can actually see it collecting data and see the data as it's sequencing the DNA. It's also very portable. As you see, it's just this here. And it also has very many applications, and it's highly used in environmental samples and agriculture. It's also in infectious disease surveillance and biomedical research. And this is also continuously improving. And it's a very newer software, so there's people that are actually using this more quite often. And yeah, thank you.